give it a couple more minutes maybe for folks to trickle in.
Oh my God. Oh my God.
There should be a sort of like a waiting list of folks that are still kind of coming into, because usually like in a lecture room, you can see if people are still trickling in. We have no idea if anyone's still at the door. So I'm just going to get us started. My name is Namik Kidlich. I'm an associate investigator at the Laureate Institute for Brain Research. And I would like to welcome you all today to our WKW Frontiers in Neuroscience lecture. It is my distinct pleasure to welcome Dr. Tanja Jovanovic to LIBR and Tulsa today. Dr. Jovanovic is a professor in the Department of Psychiatry and Behavioral Neurosciences and the David and Patricia Barron Chair for PTSD Neurobiology at Wayne State University in Michigan. She also directs the Detroit Trauma Project, which investigates the impact that urban trauma exposure has on the developing brain. Dr. Jovanovic received her PhD in neuroscience and animal behavior from Emory University, where she was also an assistant professor in the Department of Psychiatry and Behavioral Sciences in their School of Medicine for a number of years. While at Emory, Dr. Jovanovic directed the Neurophysiology Laboratory with the Grady Trauma Project, which studies the relative contribution of genetic and trauma-related risk factors for PTSD in a traumatized low SES and minority urban population. Currently, her research program focuses on the interaction of trauma, neurophysiology, neuroendocrinology, and genetics in stress-related disorders in adults and children in high-risk populations. She employs both psychophysiological and brain imaging methods to investigate these biomarkers of risk for trauma-related disorders. Dr. Jovanovic has received funding from the National Institutes of Health and also has an Independent Investigator Award from the Brain and Behavior Research Foundation. She has published over 100 peer-reviewed papers, and today we'll be discussing the neurobiological effects of trauma in adults and children. I think we are in for a real treat on this cold Oklahoma day. Please join me in welcoming Dr. Jovanovic. And with that, I'll hand it over to you. Thank you all.
Thank you, Namik. That was really an excellent introduction. I always feel humbled when someone reads that because it doesn't feel that way most days. But anyway, I'm so glad to be here and to tell you a little bit about my research. I know my title is sort of broad, neurobiology, but since you guys all understand the brain better than I do, I'm actually going to focus on things other than the brain directly. So let's see if I can share my screen. Okay. Does everybody see the full screen?
Yes, we do. Sorry.
Okay, great. Thank you. So as Namik mentioned, I am now at Wayne State University in Detroit. I moved from Atlanta where I was over 20 years, and I am just now starting to get adjusted to this winter. And I'm hearing that you guys got hit with the storm yesterday, much like we did. And I'm trying to really be very optimistic about how beautiful the everything white is outside right now. So I'm going to talk to you a little bit about the research that we did at the Grady Trauma Project. As I said, a lot of the data that I'm going to talk about was collected there. And I'll show you just a little bit of what we're continuing to do in Detroit, with the focus really on urban youth and how civilian trauma is impacting brain development. So I want to start off with acknowledging first my lab. We had several people move from Atlanta to Detroit with me, and I was very grateful for their help in starting the lab. And you'll notice we have a new lab member here sitting in his mittens and mask. But in addition to my lab members, the Neuroscience Center for Anxiety, Stress, and Trauma is also here now in Detroit. And then again, I want to acknowledge from the Grady Trauma Project, the data I'm going to be showing you today is largely due to Jenny Stevens and Sana Van Rooij, of course, Carrie Ressler, who started the project back in 2005. And then my postdoc, Anai Stenson, who is now with me at Wayne, also got her PhD at Emory and moved with me. And she's been really doing a lot of the developmental work I'm going to talk about. So we all know that trauma exposure is very common. And I like to cite this paper because it's relatively recent. And of course, Ron Kessler, who has done a lot of the epidemiological work of assessing trauma, had this paper with the World Health Organization looking at trauma exposure across the globe and looking at both low, middle, and high income countries. And one of the big points of that is that trauma is truly ubiquitous. And there's really no place on this planet where there is no trauma. And so trauma is common, but PTSD is less common than the trauma exposure. So 7 out of 8 Americans might have PTSD, about the equivalent of 8.5 million adults. And these are statistics from the Cohen Veterans Biosciences Center. And we do know that much like mood and anxiety disorders, women are twice as likely to get PTSD as men. But most of the statistics we know about PTSD is in fact based on military trauma exposure to either Vietnam Wars, Gulf Wars, Iraq and Afghanistan wars. And so one of the things that the Grady Trauma Project did was really be the first to look at urban rates of trauma. And so this was at the Grady Memorial Hospital that's downtown Atlanta. And it serves primarily a low-income African-American population in inner-city Atlanta. The thing that's unique about Grady Memorial is that you don't have to have insurance to be seen by a physician. And because of that, because it's a publicly funded hospital, it does see a pretty large number of low-income individuals, which comes along with high rates of trauma. And so in the primary care, this is not in the mental health clinic, but in the primary care, we're seeing almost 90% of the individuals experiencing a major traumatic event, and almost half are meeting criteria for lifetime PTSD. So if you think about across the general population in the US, the rates being around 10%, this is 5 times as much. For, again, experiencing a lot of trauma. And what we also have learned after now collecting data on almost 13,000 people at Grady over the years is that multiple types of trauma is the rule rather than the exception. So we typically see individuals with 2 or 3 or 4 more different types of trauma. So that doesn't indicate chronicity or frequency of any particular traumatic event, such as domestic violence, but that someone who has had domestic violence may also be more likely to be in a car accident, may also have survived a natural disaster. And then the other component is that trauma happens early, and we see really high rates of childhood, physical, emotional, and sexual abuse, and also witnessing violence between caregivers. So the way the data are collected at Grady is, again, in the primary care waiting rooms. Our RAs go out with iPad and they collect a DNA sample because Grady is really looking at gene by environment interactions in the impact of trauma. So they get and on this iPad, they'll collect a trauma history and also symptoms. And then the individuals also who are collecting data will write a summary note. And so I want to just share with you some of the case examples. So 8622 is a 33-year-old African-American female. From the age of 6, she has sustained severe physical and emotional abuse by her father and repeated sexual abuse by her stepbrother. She was unwilling to talk about any of her traumas, but has significant symptoms of PTSD. She is also drinking about a 6-pack a day and is currently using cocaine. She has no close relationships with any of her family members. She has no friends, nor hobbies, nor is she interested in leaving the house. Her current boyfriend has been taking care of her since her last suicide attempt a few weeks ago. This was her 3rd attempt. So I wanted to show you just as an illustration, this is a very typical, unfortunately, example of what we see at Grady. And one of the things that it illustrates is the high levels of comorbidity that we see. So we see a lot of substance abuse and suicidality, in addition to PTSD symptoms and trauma. So in thinking about some of the risk factors for PTSD, and particularly thinking about gene by environment interactions, this is the very simplistic idea of how one might think that genes and environment interact to result in PTSD. But as we know, the picture is not this simple. And there's not a single gene that could lead to PTSD. And in fact, now we're seeing not just multiple genes, but in GWAS studies of, the whole genome, we're looking for gene by gene interactions as well as polygenetic risk for PTSD. And then the environment, the trauma, is not a single event that happened that we can easily isolate. It's typically chronic, it's early, it's repeated. So we don't have something very simple to think about as this environment. And then PTSD itself, again, is a messy phenotype. It is comorbid, as you saw with so many other disorders. And so it's hard to isolate. And in fact, PTSD as a disorder is heterogeneous with many different symptom presentations. So one of the things that might be really useful in the way we think about it is to focus on the brain and look a little bit more closely at neurobiological phenotypes or biomarkers. And for those of you who are deeply steeped in NIH, you might use RDOT constructs as terminology to think about some of these phenotypes. And I'll kind of use the word biomarker and phenotype, and I'll tell you in a little bit how I define this specific word. But if you look at putative biomarkers for PTSD, there are many different options and many different systems, not just the brain, but other organs in the body that are affected by trauma. And so HPA axis, MPY, sleep patterns, heart rate and heart rate variability, inflammation, hippocampal volume, and the PICAP system in the PAC1R genotype are just some of the different phenotypes that people have linked with PTSD. But the one that I'm going to talk about today is really what we've been calling impaired fear inhibition. And one thing that we found about impaired fear inhibition, that it is often associated with many of these other biomarkers as well. So before I talk a little bit more about that, I want to specify what I mean when I talk about neurobiological intermediate phenotypes. And this is different than perhaps an endophenotype, because an endophenotype by definition is heritable. And I don't think that an intermediate phenotype needs to be heritable to be useful. But what I think that makes it useful is that it should be related to symptoms of the disorder. It should be related to the underlying neurobiology of the disorder. And ideally, we should be able to model it in preclinical or rodent studies to have this translational approach and to be able to look a little bit more at mechanism. And so one of the ways that PTSD has been studied is looking through a fear conditioning model. And this is where that preclinical approaches and translational approach is really important, because we know a lot about fear conditioning from rodent data going back decades. And so as Carrie Ressler always used to say, fear conditioning and the fear system for PTSD is very low-hanging fruit, because it's one of the things we understand the best. And what we know is that the brain circuits involved in fear and threat responses are also the ones that support fear conditioning. And we can think about the onset or etiology of PTSD in sort of very rudimentary terms as a fear conditioning event, where a conditioned stimulus, such as, like in this case, a car, gets paired with an unconditioned aversive stimulus, such as this car accident or trauma that's happening. And those things then become associated at the level of the brain. But one of the things we need to start off with is really an objective measure of fear. And this can be a little bit problematic. And I'm gonna show this cartoon from the far side. You know, it's the best cartoon for any research talk. But we all know that, you know, dogs anecdotally can tell when people are afraid. And so what we really need is like a ferosensor that maybe the dogs have for sensing fear. But the way that we've done it in our lab is using the startle response. And again, this capitalizes on this rich history of work that's been done in rodent models. And Mike Davis, who was one of my postdoc mentors, really did a lot of work in this field of fear potentiated startle in rodents, where you can measure their startle response by how far they jump in this box. In humans, we do it by measuring the eye blink contraction to a loud sound. And so in our laboratory, we use fear potentiated startle. And so our rodent box looks something like this. It's a lot bigger, but we can measure that eye blink contraction again to a loud sound. And we're measuring it with those electrodes under the eye. And then we're also capturing skin conductance and heart rate. But the other things I want to point out in this image is that we have this vest that's sitting on the chest. So this is a modified camelback. Most of you have probably seen it, especially any avid bikers in the group. The way that we use it is to deliver through this tube a pressurized air blast to the throat. And so this is going to be our unconditioned or aversive stimulus. So in our studies, we have not used shock. We piloted it a while ago, and we found that air blast was very effective in potentiating the startle response. And it was a little bit easier to talk about and recruit people into our studies, especially dealing with patients and children, when we didn't say we were going to shock them. So they get the air blast. The other things that I want to note is that there's a computer monitor where the shapes are going to appear on the screen that are going to be our conditioned stimuli. And then we also have a response box, and on each trial, the participant is going to label whether or not they're expecting an air blast on that trial. And then lastly, you see the headphones. That is where the sound is delivered. So the sound is delivered on every single trial. And so this is what the fear acquisition session looks like. There are 3 blocks. Each block has 4 different events of each trial type. So we have a noise alone. Sometimes you'll see in startle studies, this might be called the ITI startle response. It's really there to capture how much you startle to the sound itself. Then there's a startle, again, the sound presented during a CS+. So you'll see the design here for 6 seconds. There's a shape on the screen. Then there's a startle probe. Half a second later, they get the US or the air blast. On the CS- or the safety trials, you'll see the shape. There's also a startle probe that's again measuring the eye blink response to this shape, but no air blast. And we use during acquisition 100% reinforcement. And so just to show you first what the US expectancy is. So an expectancy rating of 1 is that there's a maximal expectation of an air blast on that trial. and -1 is that they don't expect this. And you see across the 3 blocks of acquisition, this is in trauma controls, we see very nice, almost perfect discrimination, and it's already there early on, and then it's almost at 100% in the later blocks. And looking at the PTSD group, it looks pretty much the same. So we don't see any patient-related differences in US expectancy, in that individuals are very clear that when they see a shape come up on the screen, that they are expecting the air blast on a CS plus trials and are not expecting it on a CS minus trial. When we look at fear potentiated startle responses, and these are startle responses specifically to the cue after subtracting the noise alone trials, you can see in the controls that it takes about a couple of blocks to start discriminating. So we start to see it in block 2, and then it gets stronger in block 3. In the PTSD group, it takes much longer to get to this point of discrimination. And in fact, even at the last block, it's only at trend level in this sample size. If you look at the end, you can see the biggest difference in the group is the magnitude of fear potentiated startle. And it's there really for both trial types, the CS plus and the CS minus. But the effect is stronger for safety. And if you look at this blue bar here for PTSD group, how much fear is there for the safety is about the same amount of what we're seeing in the controls for the danger stimulus or the CS plus. And then we wanted to see whether this was in fact related to symptoms of PTSD. And so when we look at a correlation, we found that, and this was back when we were using a DSM-4, so we had the 3 symptom clusters. The fear potentiated startle to the CS plus or the danger cue was correlated with re-experiencing symptoms, and startle to the safety cue was associated with hyperarousal and re-experiencing symptoms. Notably, skin conductance had very little response to association with any of these. And in fact, when we do a regression where we control for age, sex, race, childhood, and adult trauma, only the startle to the danger cue is still significant for re-experiencing symptoms, and startle to safety cue remains significant for hyperarousal symptoms. And skin conductance is no longer associated with any symptoms. So not only is startle or this fear potentiated startle to safety, and this is what I'm going to talk about as fear inhibition. It is the ability to reduce the amount of fear to a safety cue that's a known safety cue, because remember that our participants, almost all of them understand that they're not going to get an air blast on this trial, but they're still showing heightened fear responses at the physiological level. So it is this inability to regulate that fear that we're calling impaired fear inhibition. And so we recently used this paradigm in a treatment trial, and this was a pharmacological trial using a CRF type 1 receptor antagonist with a GlaxoSmithKline drug. And looking at pre and post treatment, we did see that the individuals who were on the drug, and this is a randomized clinical trial, improved their fear inhibition after treatment, whereas the placebo group did not and actually got slightly worse post-treatment. And so the reason I show you this is really just to show you that this is modifiable target and that fear inhibition can change with treatment. So again, thinking back about phenotypes and ARDA constructs, I think this is something that is plastic enough that provides a really nice treatment, both a tracking treatment outcomes, but also potentially a target itself. And this I just want to show you, we've now looked at fear inhibition in several different PTSD populations. We've looked at combat civilian PTSD and in individuals who have depression with PTSD. And in all of these groups, we see that if they have a PTSD diagnosis, they're showing impairments in fear inhibition. We've used extinction, simple discrimination, and conditional discrimination. And again, in all of these paradigms, we are seeing this impaired inhibition response. This paper, this review paper with Sana that just came out a couple of years ago, She actually, as part of her dissertation, used a stop signal task and found again that even at the level of the brain, just looking at either motor inhibition, response inhibition, you can actually see a lot of those same phenotypes for PTSD related to both risk of developing PTSD, but also maintaining PTSD through treatment. So how is this related to the brain? So we, again, can capitalize on what we know about fear inhibition in the animal literature. And we know that a lot of the prefrontal cortex is inhibiting that amygdala response that's driving the fear response. So what we wanted to do as part of the Grady trauma population is come up with a task that would engage the ventral medial prefrontal cortex in our Grady individuals. And although at the time there had been already several studies, Mo Milad had been doing some conditioning and extinction tasks in the scanner, but a lot of those were engaging prefrontal cortex either during extinction or extinction recall, which was 24 hours later. And we found this to be potentially very challenging for our population. And we wanted to do something that would minimize burden, that we wouldn't have to have a lot of exclusion criteria because we really wanted to be able to generalize the findings. So we picked a task that was really simple and straightforward. And so we just used a go, no-go task, again, regardless of whether we were looking at fear circuits or not, we just wanted to get something that we felt could pretty reliably engage the VMPFC. And that is what we found, and we found a great group difference, so that those who had a diagnosis of PTSD compared to trauma controls without PTSD did show a lot less engagement of the ventromedial prefrontal cortex. But the other thing that was important is that we did find an association with fear potentiated startle, specifically again to the CS minus. And we found a negative association, so that the higher the engagement of this area, the less fear they were showing to the CS minus, which is the appropriate level of fear inhibition. So we were seeing good inhibition with strong engagement of this area. So coming back to how I was defining my neurobiological intermediate phenotype, related to symptoms of disorder, we did see that with fear inhibition. Related to the neurobiology of the mental disorder, we did see that with impaired fear inhibition. And then again, we can model it in preclinical studies because that's actually where we started with fear conditioning. So I want to tell you a little bit more about our recent work during development. And we know that trauma during childhood is one of the greatest risk factors for developing PTSD in adulthood. And a lot of this data is based on retrospective reporting from adults. Even in our Grady sample, our average age in adults is about 40, and we're asking them to report on their childhood trauma. And this can be really unreliable and difficult to pinpoint. And what we know, especially in recent times, Katie McLaughlin's been talking a lot about different types of trauma, and that childhood adversity can come in different forms, and in fact can have very different effects of the brain. And so she's been really talking about adversity dimensions and thinking about threat and deprivation as very different dimensions. And so abuse, physical abuse, violence exposure, we might think about as threat, but neglect, poverty, that might really be deprivation. And so in this recent review, she really looked at a meta-analysis of many different imaging studies, and looking at the medial prefrontal cortex and amygdala, the impact of threat really was to reduce amygdala volume, but increase amygdala activation, decrease medial prefrontal cortex, and then there are mixed findings about connectivity between the two. And if you look at deprivation, you see some different patterns. You don't see this high impact on amygdala activation, this positive impact on amygdala activation. And so one of the reasons why there might be these mixed findings is not just that trauma type is important, but timing during development is also important. So this study, Dylan G in Nim Tottenham's lab published back when she was in Nim's lab, was really looking, and this was a fearful faces task in kids across a pretty wide age range, and looking at amygdala MPFC connectivity, they saw that that connectivity was positive early on in development, but around age 10, really shifted to a negative connectivity. So that in adulthood, we see this really strong negative association between activity in the prefrontal cortex and the amygdala. Again, this was during the task of showing fearful faces. So this seems to be what we're seeing in adulthood, but this is the developmental pattern of how we might get there. So we wanted to do a similar thing with our population using fear potentiated startle. And so we have a second booth in our lab where we do a very similar experiment to what I just showed you. We reduce the air blast a little bit, but the air blast is very well tolerated in our children. And this is really important in doing fear conditioning studies with developmental cohorts like this or pediatric populations, because you don't really want to use electric shock if you don't have to. Danny Pine's group has used the screaming lady, and a lot of their experiments have found that some of the kids have a hard time tolerating the screaming lady. And if you've ever seen it, it's very scary. And even some adults have a hard time making it all the way through the experiment. So the goal really is to find a stimulus, a US, that is aversive enough so that it'll potentiate the fear response and potentiate startle, but is not so aversive that individuals quit early, don't want to come back. In our developmental work, we're looking at longitudinal design. So it's very important for us that the kids leave the lab feeling okay about the study and want to come back when we call them again. So again, we can look at eye blink responses, and this is showing you the 3 blocks of acquisition, and you see, much like adults, we see really nice discrimination by the end of acquisition. But if we break it up by that age group, again, using age 10 sort of as our cutoff, you'll see that it's really the older kids that are showing this really nice learning pattern. So this is the end, this block 3. And if you look at discrimination, this is CS plus minus CS minus, the difference between the two, it is significantly larger in this older age group. So this was in the Grady trauma population. So we also knew that these children have pretty substantial levels of trauma exposure. So we wanted to measure that. And what we used was the violence exposure scale, or the VEXAR. And this was originally developed by Nathan Fox and group back in '95. And we worked with Nathan to get some of the data from the Grady population. So what we found is, first to just describe the measure, I think this is important that it's really a cartoon measure. And it was designed for a younger cohort than what we're studying, but it works really well in our age range, again, which is about 8 to 13 altogether. And the way it works is that there's a child to the side, and this child's name is Chris, and Chris can be a boy or a girl. So there's a girl and a boy version. And the way the question goes is, Chris sees a person beat up another person. And then they look at the thermometer and you ask them, how many times have you seen a person beat up another person? So we do that with a couple of different questions. And so they respond on a Likert scale. And this is just showing you how many kids report at lots of times having experienced or confronted with different trauma types. So you'll see very high rates of witnessing assault, witnessing arrest. But even again, at the age of 8, about a third of our kids have been confronted with murder. and assaulted or witnessed drug trafficking. So it's a very high level of trauma exposure. And I want to share with you again a case example from the study. 119110 is an 11-year-old African-American male. He is one of 5 children, and his mother, who is 29 years old, is currently pregnant. He reported that school is very important to him and he has goals to graduate and get a good job. On trauma measures, he reported witnessing serious physical fights between children and adults, witnessed a stabbing, neighbors arrested, and seeing a dead body in his community, a gunshot victim lying in the street. He endorsed many symptoms of PTSD, including frequently watching out for danger, avoidance, and having frequent memories of the event. And again, this is unfortunately not a rare occasion in our sample. So looking at trauma exposure, PTSD, in our mothers and children, we did find that, and as you would expect, there's a strong correlation between child-reported exposure to trauma and, again, child-reported symptoms of PTSD. So we wanted to see how this relates to fear inhibition. So this is looking at Startle to the CS minus. And again, we see that age effect, that this, again, if they're showing appropriate fear inhibition, it should be low. So here we're seeing in the older kids, we're seeing that response. And this was in the low trauma group. In the high trauma group, we are starting to see in the adolescents some reversal of that, that they're showing with high trauma increased fear to the CS minus. It's a little bit interesting that the younger kids are actually showing better inhibition with higher levels of trauma. And we don't know whether this is some form of early adaptation to the environment. We have seen similar things in, again, some of Dylan G. and Nim Tottenham's work with the adopted kids from Romanian orphanages, the post-institutionalized kids, with showing some potentially accelerated or learning with early adversity. And so we don't yet know what this will look like as we continue following these kids. But if we look at the adolescents, we do see this positive association between not just trauma exposure and fear-potentiated startle, but also PTSD symptoms and fear-potentiated startle. We didn't see this association with the younger kids. And then one of the other things we wanted to see with respect to whether this was a risk factor was to look at it in a longitudinal study. So again, following these kids later, but we also wanted to look at the impact of this PAC1 receptor genotype, which has been associated with PTSD in adults, including startle responses in adults, specifically in women. So in this study, the CC genotype represents the, quote, risk genotype for PTSD. And this is visit 1, and then 1 year later, we had a follow-up visit, and this was about 100 kids later. And again, looking at the CS minus or the blue bar here, you'll see that this risk genotype is increasing startle to both of the CSs, but much more so to the CC genotype. And so we found an interaction with visit when we look at just that second visit and we break it up by violence exposure on the VEXR. Again, it's the CC genotype combined with higher violence that's showing this impairment. And so we get to see this very significant difference between CC and the G carriers in the high violence group, but not in the low violence group. And so this study is what we're now continuing in the Detroit Trauma Project. We're calling it the Critical Period Study, and that's just because that was the name of the grant. And we just have really started now with the baseline visits in Detroit, but I wanted to just show you how the trauma exposure compares between Atlanta and Detroit. And for the most part, they're very similar. We're seeing a little bit less of a frequency of witnessing arrest and witnessing assault. But for the most part, we're seeing some of these really high rates of trauma. And then looking at fear inhibition in the brain, we again used a go, no-go task with kids. And one of the things that is notably different about this task than the one that we used in adults is that this one does in fact incorporate a fear stimulus as well. So this is based on a NIMS task that she originally developed. And what that allows us to do, like in this case, the go stimulus is the neutral face, the no-go is the fearful face, and then in a second run, it switches. But we can look at both response inhibition to no-go versus go trials, but we can also look at fear no-go versus neutral go trials. And looking at the ventral medial prefrontal cortex, we actually saw a positive association with increased trauma exposure. So this was a little bit surprising. We expected that increased trauma exposure would actually be reducing activation to this inhibition trial rather than increasing activation. But one thing to note about this sample is that we actually had pretty low symptoms of PTSD in this group. We did, however, find that negative association with fear potentiated startle, which is what we also found in adults. And in adults, that was the whole sample, not just the PTSD group. So again, we found that ventral medial prefrontal cortex activity was higher in individuals who are showing better fear inhibition or lower startle to the safety signal. And again, as I noted in this sample, especially the imaging subset of the sample, we didn't have very high rates of PTSD. But I wanted to show you this paper actually that just came out. Again, this is now out of Dylan's lab, where they did look at condition inhibition in the scanner, looking at children with and without anxiety disorders. And looking at inhibition, this was, again, looking at a condition inhibitor stimulus versus a novel stimulus. So here, you want higher activation to this inhibition stimulus. And in fact, the children without anxiety disorders, here shown in orange, showed more activation to the safety signal. And that was also found in skin conductance responses or physiology. So you'll see that the anxious children showed across the board more arousal to all the stimuli compared to the non-anxious children. So I think this again goes back to showing that fear inhibition can be really a marker of not just symptoms, but also what's happening at the level of the brain. And just with a few minutes that I have left, I want to come back to thinking about the impact of the caregiver in our families. And one of the things that we've noticed, and I've talked to some of you guys about today, is thinking about intergenerational effects. And this was, again, started at Grady, and we're continuing to look at it in Detroit, thinking about moms and kids with trauma exposure. So Anais Stenson took a lot of the dyad data from Grady from the Mom Kids Project, and she did a latent class analysis where she looked at maternal lifetime trauma exposure of all types, also all her childhood trauma exposure and PTSD symptoms. And when she did a latent class analysis, she came up with 4 different classes of moms in maternal trauma. And the high trauma and PTSD moms, their children, so this is Startle in the child maternal class of trauma, their children had much higher fear to the safety signal than either moms with moderate trauma or moms who had high levels of sexual abuse. One of the things you'll note is that the moms with high sexual abuse also had children with blunted fear responses as well. And some of that we are finding in even adults and adolescents with high levels of sexual trauma, we're seeing blunted fear responses. So this data actually was analyzed co-varying for the independent effects of child age and their own trauma exposure. So these are results that point specifically to maternal factors in predicting child startle. And one of the things that we found with some of our other measures, that some of the mechanisms for this, and again, will include parenting, but also we're looking at some of the biological effects, looking at both genetics and epigenetics. But one of the things we're seeing about parenting behavior is that parenting stress is mediating a lot of these intergenerational effects. And moms with higher trauma and higher PTSD are also more stressed out by parenting. And the more stressed out by parenting they are, the more their children are showing both symptoms and psychophysiological these dysregulations. But then on a more positive note, we also see some effects of maternal buffering. And so several years ago, again, coming back to Dylan, I was in a session with Nim, and Nim was presenting this data. Again, this was a long time ago that we were in this session. And she showed this data, again, looking at amygdala MPFC connectivity in children and adolescents. And she found that if children were looking at a picture of their mother, they were actually showing better negative connectivity than if they were looking at a stranger. And you didn't see this effect in adolescents because they were actually already showing good negative connectivity. And so when we were at the session, I went back to Grady and I realized that we've been doing this sort of informally, where we've been thinking about intergenerational effects where we startle mom and kid. And so we have two booths like this back to back, and the parents get startled in booth B and kids get startled in booth A. Now, as it happened at the time, there were several occasions where mom, for some reason or another, didn't get startled. Sometimes it was because she was maybe pregnant or at high risk, or maybe she had already been startled at an earlier visit. And so in about half the kids that we had startled, half of them, the mom actually was doing something else while the kid was being startled. And what she was doing was going to a different room to complete some of the interviews. And so the kid would see the mom leave the room, or the child would see the mom go into the booth next to her. What's important to know is that there's no sound or visual connection between the two booths. So mom goes into the booth, can't talk to the kid during the session. So I just wanted to show you this picture in this context. So this is my daughter who got startled. This was when she was 9 years old. And this is one of the downsides of being raised by a neuroscientist, is that you have to test out all the experiments on yourself first. So what we found is, again, in the younger age group is what we had seen already, that we didn't see discrimination, but we did see it in adolescents. However, if the mother was in the booth next to the kid, we were seeing that discrimination. So again, it's not direct learning. There's no instructions. They don't have an opportunity to talk to the kid prior to this experiment because they haven't done it yet. So just having the mother in the booth next door is potentially impacting this fear inhibition. And we've looked a little bit about what might, and again, this is a relatively small sample size. This is 100 kids altogether. So this is something we want to look at a little bit more, but it was really an interesting, and in this case, a very post hoc observation. So, but it did seem optimistic. So I think with that, I'm going to go ahead and summarize. So I hope that I've shown that impaired inhibition of fear can serve as an intermediate neurobiological phenotype or biomarker for PTSD. We see greater fear potentiated startle to the CS minus, or the safety signal, is associated with PTSD symptoms in adults and also adolescents. Fear potentiated startle to safety increases with trauma exposure, but only in adolescents, and again, associated with PTSD symptoms. Ventral medial pre-FC activation during any inhibition task, so just a go, no go task, was associated to fear potentiated to startle, and this is at 2 different sessions, so it's not concurrently measured, but we see that in adults and in children during development. And we see some buffering or facilitation of fear inhibition in younger children with parental presence. And again, with adolescents, we didn't see that effect because much like the buffering effect that Dylan saw, adolescents are already performing well regardless of where their parents are. So with that, I want to thank all of you guys for listening. I want to thank our funders that make it possible for us to do science. And I also want to show you my daughter here, who is now a sophomore in college and has survived all of her fear conditioning experiments unscathed, hopefully. So I'll be happy to take any questions.
Thank you, Tanja. So if folks want to jump in and ask questions, go right ahead, or if you prefer to post in chat, I can read those out as well. So
I can go ahead and stop sharing.
I have a question and a comment, Tanja. Thank you very much. It's a very nice presentation. It was a pleasure to listen to it. Being the fact that fear inhibition impairments can be proposed as a biomarker of PTSD, I wonder how much that same trait could be like a window of opportunity to treatment, in that some people have proposed inducing fear as bringing traumatic memories to a labile state and then inhibiting its reconsolidation. If I would like to hear a comment on that, if you can. Thank you.
Yeah, that's a really good question. So, I think some of the preclinical work with Marie Monfils that she used this was in a science paper several years ago now, where they did exactly that. They reactivate the CS plus sometime after fear conditioning and then wait some amount of time. And the amount of time you wait is really important because that's when the plasticity window is open. And during that time, when this window is open, then you do extinction on the CS plus. And the idea being that there's something important about reactivating that memory. And so in this experiment, which I thought was really quite sophisticated, what she did was show a CS plus, wait some amount of time, and then again, repeat the CS plus during extinction. And in the control condition, the only thing that was different was that timing between the initial presentation of the CS plus and then the extinction. So that if you just do extinction, you don't have that plasticity window. So you're maybe not targeting that reconsolidation time. So, some of that data, Danielle Schiller did a similar experiment in humans and found the similar effect of reactivating that CS plus. We did it in healthy humans and found that it sometimes works and sometimes doesn't work. And so I will say that in at least the human experimental literature, this has been kind of a mixed finding as to whether this reactivation and reconsolidation can be made to be labile enough. But we did a study with Barbara Rothbaum, and this was in a fear of flying cohort. So Barbara Rothbaum has really pioneered using virtual reality for exposure therapy. And the way that we did it was in patients who had a fear of flying, they were shown a virtual airplane. They're either in a virtual airplane or a virtual office. And then that was used as the reactivation CS. And then they went into exposure therapy. And the exposure therapy, again, was for fear of flying using virtual reality. And then we compared outcomes for the people who saw a neutral stimulus prior to exposure therapy versus those who had seen the airplane stimulus before. And clinically, it didn't really make a difference. So the problem with exposure therapy is that it's actually tremendously effective for phobias as well as PTSD. And so most people, their symptoms decreased regardless of this first trial. We did see some differences in physiology. I believe heart rate was a little bit lower in the people who saw the reactivation cue. So potentially there's something there, but I will say that we had to kind of dig and maybe squint to find that. So I think that that was a very promising initial line of research because it was a non-pharmacological potential intervention. One of the problems with extinction is that if you think of the trauma as the conditioning, then you can't really do much about the trauma for people with PTSD. It's already in the past. And this was a promising idea that if you could make that consolidation window open again, you could potentially manipulate that in some way. But I don't think those memories and consolidation is as labile as it might seem.
And Tanja, how about sleep? Usually we take nightmares as a clinical feature of PTSD, but I wonder how much those same nightmares in certain phases of sleep might also have a role in reconsolidating on a day-to-day basis the traumatic memories and all the arousal associated to it. So do you think there might be a window of opportunity to intervene in sleep, just like in depression, where you have sleep deprivation for the second half of the night and you bring about an antidepressive response?
I've never thought of that. That's a really interesting way to think about it. I guess you would have to know when those nightmares are going to happen. And I don't, it would have to be, I guess, one of the principal symptoms that the patients are presenting with, and not everybody experiences nightmares, and certainly maybe not frequently enough that you could build a study predicting those events, unless you did, potentially you could do it in either looking at sleep measures at home, maybe using actigraphy or some other maybe ambulatory or watch-based methods where they coded experiencing nightmares when they were happening at home. But that's a really interesting idea. I hadn't thought of that as a potential reactivation trial.
Tanja, it's Robin. Great talk.
Hi, Robin. Good
Hello.
to see you again.
Good to see you again too. So I have several questions, and we're going to meet later. So I don't necessarily need to go over all of them right now. But one thing I was wondering, because I have always been intrigued by the whole mother phenomenon as well, in terms of amygdala reactivity and connectivity and all of that. Have they done any studies in adults looking at like a social support person, or even, because I'm thinking like in a therapy relationship, once you have rapport and a connection, that actually the therapist might be serving in that role to help enhance inhibition? But has any studies been done in adults to see if you can get the same for people who have impaired inhibition, to see if you can get similar effects?
Don't know of any people who have looked at it in impaired inhibition. I do think there are some studies of partners where you either, in a similar way, maybe you're showing the partner's face in the scanner, but I really can't come up with an actual study. I think this is like faintly somewhere in my mind, but I do think that would be a really interesting thing. I will say that one of the problems with thinking about safety signals in therapy is that they sometimes become a problem. So if you become dependent on your safety cue, so whether maybe it's a service dog, maybe it's another person, maybe it's the therapist, and then once you no longer have that cue around, do all your symptoms come back? And so in therapy, it might not be directly applicable to translate the safety signal per se to a therapeutic. or it may be, but in a temporary way that's then tapered off in some way. So
Yeah, that's what I think anyway, my take would be that we do that in an informal way without meaning to, right? In terms of providing that support, even if we're not doing graduated exposure, that we're still providing some sort of support in a safe environment, to begin with, to try to that might enhance their ability to actually manage their responses.
Because I think with exposure therapy, it is very effective, but there are high rates of dropout. So it's effective if you stay till the end.
Yeah.
And potentially where fear inhibition might actually provide a good role is to keep people in therapy. So maybe if you can reduce some of the fear, they might stay till the end, and that means they get better. Again, the caveat to that is that you can't blunt the experience, because we see that either individuals who get benzodiazepines right before exposure therapy or are not engaged in exposure therapy, even on a physiological level, don't get better as much as those who actually engage.
Yeah, kind of a physiological rationale for why the common factors and rapport and all of that might be, is so crucial, even with, say, exposure therapy, that if you don't have that to try to help support them through it to begin with, then they're just not going to be able to engage. Anyway, it's just an interesting, I think, take on kind of bringing those two together. So I think that's awesome. And then the other question I had, do you feel like, say, the reduced inhibition for people who are still in volatile or dangerous environments, to have kind of reduced inhibition and reduced safety signaling in some ways may be adaptive, that, identifying even ambiguous cues as potentially threatening and things like that in some ways may be adaptive if they're still within that context?
And I think we've thought a lot about that in thinking about civilian versus a military trauma, because in military populations, usually at the time you're seeing them, the environment is no longer dangerous. So the, really fear inhibition is or showing impaired fear inhibition is not adaptive. So we've thought about whether, if you leave the laboratory and you go back to your neighborhood where there's a lot of violence, is it actually better to show high fear responses across the board and generalize? And I think that it is not, in fact, adaptive, because what we see is this high rates of hyperarousal that can be really impairing. So I think what's very adaptive is to show high levels of fear, to appropriate fear signals, and to be able to discriminate between danger and safety. But I don't think it's adaptive to show fear to known safety objects. It's just that some are not so known.
Hi, it's Bart. So along those lines, do people that have more fear arousal, are they also perhaps less likely to try to change their situation? Are they also afraid to leave? Are they afraid to, make the changes necessary that they need to get themselves out of the traumatic situation?
Yeah, I think for sure, we see that. And so I think one of the symptoms that is most associated with this laboratory paradigm of impaired fear inhibition is this hypervigilance, which is like constantly being on alert, constantly being vigilant. And I do think it makes it very hard to, change situations. And granted, in a lot of our studies, when we're looking at kids, they just don't have that possibility. But even in women who are in domestic violence situations where potentially, they could leave that scenario, there are so many barriers to that it's very difficult.
Any other questions? I had and then we can wrap up. I had a quick question about, we've actually, well, Robin and I have talked a little bit about this, about finding ways to sort of enhance that fear inhibition and finding ways to enhance responses to the CS plus. And by enhance, I mean sort of inhibit them, meaning enhancing the differentiation. And so we thought about perhaps using something like self-awareness exercises or emotional awareness exercises, so something via mindfulness, sort of bringing attention, an increasing ability to bring attention to then during an extinction process be able to recognize a difference of what a CS plus and CS minus represent. And so I was wondering if you know either studies or what your thoughts are on using something acute prior to perhaps extinction training that could help differentiate CS plus from CS minus with the hopes of reducing reactivity to, well, either perhaps over time,
Yeah. So I have two answers to that question. One is the way we've done it, and we've only done this, I think, one time, is that we did a study where there was no response keypad during acquisition. So you didn't label every trial as either expecting the air blast or not. And if you remove the labeling on a trial-to-trial basis, at least, you do get less discrimination. And I think it's that they don't pay attention to the cues as much. They're not having to label them. Now, you still actually get significant learning. And one of the things that we found is that, you know, because fear learning is so necessary for survival, it is really hard to do anything to remove fear learning. It's so preconscious, Mike Davis used to say, even a turtle without a cortex can learn to be afraid. I think it's just so adaptive. But fear inhibition is actually much harder. So fear inhibition does require some cognitive input and awareness of the stimuli, paying attention to what's going on. So we see a little bit less discrimination when we remove the keypad. One of my collaborators, and I don't know if you guys know Matthew Price, he's actually at the University of Vermont. His K award was doing that. So I don't remember exactly what the paradigm was that he used between acquisition and extinction, but his goal was to use something along the lines of a mindfulness task or attention task to facilitate extinction. And if I'm remembering correctly, it had no impact on extinction. So, but I think it's important what you choose as your control. I think sometimes, you want to be really rigorous and choose a control that has maybe a milder impact or some kind of sham effect. And it may also be having some effect.
Well, thank you so much for taking the time to speak with all of us. I know you also have a full afternoon, so we'll let you get a break. Thank you all for attending, and see you at the next one.
Great, thank you.
Okay. Bye.